designed in a way that if I have a 10-bit barcode, I would have a total of 10 different kinds of readout sequences. So if this RNA has a 1, 0, 1, blah, 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 no, not blah, blah, 0, 0, 0, 0, 1, then what gets onto this RNA, the collection of encoding probes, would only have readout sequence 1, but not 2, readout sequence 3, but not the rest until readout sequence 10. So this way, I physically convert the barcode into collection of readout sequences. And then we detect the presence of these readout sequences by complementary fluorescently labeled readout probes to detect which readout sequence is present. And then, as I said, this is I already explained that it scales very well. So then, why error robustness is important? That's because even though I mentioned that single molecule FISH is very accurate, means that if I image a single gene, I could have 90% or 95% or 97% accuracy, which also means per bit I have that high accuracy. So in other words, I only have like a 3 to 5% error. So that normally is not a problem. But if I have to actually identify this molecule, let's say with 15 bits, then the error is actually at 3 to 5% times 15. So all of a sudden you have like a 50% error. So if I tell you, look, I give you a transcriptome-wide characterization of this thing, but I have 50% misidentification rate, you probably wouldn't like it. So because of that, we actually devised ways to actually do error-robust barcoding. And in a nutshell, it's a barcode-capable scheme that allows us to detect and correct error. The idea is also very simple, and we borrow this idea from a very distant field of digital communication. So when you have your cell phone text messages and so on, those are all transmitted by binary barcodes. And I still remember when I first worked on computers, I often get garbled messages and like emails and so on. These days you don't. And one key thing is because the coding schemes has error detection correction capability, which means that out of the all binary barcodes, you don't use all of them. You use a subset so that between pairs of valid barcodes, multiple bits have to flip. So in that case, if a single bit actually accidentally flipped, it will generate a message or barcode that's meaningless because it doesn't correspond to any gene. And not only that, it will be uniquely close to one valid barcode if the number of barcodes that has to be flipped is more than 3 or more. It will be at least 2 or 3 errors away from another valid barcode. So with that, I can even correct that. And that allows us to actually do high accuracy detection. So with that, that's why the name is multiplexed error robust FISH or MERFISH. So in the first MERFISH paper, we've already demonstrated genome scale imaging because I told my students that if we use this approach to image 10 or 20 genes, it's really not convincing to people that it actually is scalable to thousands. So I set this goal of genome scale imaging in our first paper, so it actually took us multiple years from the conception of the idea to experimental implementation. I'm going to skip that. But the idea of it is really allow us to pretty much image a random number of genes. So this is pretty much the same idea, the same MERFISH design, and we image 10,000 genes, and then here each individual dot, again, is a single RNA molecule. And then they're color-coded by their genetic identity, and you can see that indeed they have very non-random spatial distributions, which won't be the topic of today, so I won't go through that. But before I move on to some applications, I will say that you can all pretty much image an arbitrary number of genes, but that's only true by a number. That's not true by some other criteria, which I will get to later on to show you how do we actually get to the whole transcriptome imaging, okay? So, as I said, one of the major applications of this is indeed what we call the cell type identification and mapping, because now I can just take an intact tissue. I no longer dissociate the cells from the tissue or extract the RNAs from the cell. I just have an intact tissue that I'm going to do MERFISH. I'm going to image these individual cells and get their gene expression profile and identify their cell types. And then this way, I not only identify how many different types of cells are there in this organ but also how are they spatially